Okay, the rest of the section is going to go through basically some terminology that has to do with the whole mitosis process, and honestly the majority of it has to do with this term here, genome. And actually I'm going to suggest that you go just pause the video real quick and copy all of these terms. Copy all these terms, make sure you know them. They're all terms that are very important to this chapter, so just pause real quick, write them down. I guess you just unpaused, I don't know. But make sure that you can define those terms. When this is all said and done, either go look them up now or after the video, make sure you can write down good definitions there. So, like I was saying, the majority of the chapter has to do with terms, or this section of the chapter is going to introduce terms that have to do with this genome idea. And again, if I go back to the goal of mitosis, where we had this like squiggle that represented the genome, and if we're going to do mitosis, we need to make sure that that squiggle ends up in both of the daughter cells. So we had to make a copy of the squiggle, anyway, the genome, the information. Then that is what a lot of these terms have to do with. So let me get rid of this. First of all, when we're talking about that genome, which is the DNA, DNA takes a couple different forms, and so I'm going to pull a quick analogy here. DNA can either be, it is always made of chromatin, this term here, chromatin, but it can be described, as it often is, as chromatin, or it can be described as a chromosome, which is another form that it takes. So what I'm going to do is a quick analogy here. Let's just say that DNA is like fishing line, okay, and fishing line, and it is kind of that way. DNA is a long string of letters in a row, a molecule. It's a very long string. So here's some fishing line. Just found this picture on the internet, but my guess is this is probably like, I don't know, 6 or 7, maybe 10 feet of fishing line that got all tangled up, and so somebody just bunched it together in a very unorganized way and threw it in the grass here. But this DNA, if it were DNA, is taking up a tremendous amount of space for the amount of actual material that's there. If I compare that to, if I take that same amount of string, actually quite a bit more, if I organize and put it on a spool like this, this shows that this is 1,275 yards of string, okay, fishing line. That's a whole lot of fishing line, and it's taking up, honestly, it's taking up less space than it did on the other picture here, and which is pretty remarkable. And all we did was organize it. So we have our organized form right here, and our really sloppy form right here. Well, this form would be the chromatin. Okay, and I'm going to call it chromatin because there's another term on the list there, chromatid, and I think that they can get confusing if you say them the same way. So chromatin, and chromatin is the form the DNA is in most of the time in a cell, where it's uncoiled, it's unorganized, but the advantage of chromatin is, if you remember, everything in this is information. And when it's uncoiled, the information is spread out. There's lots of space in there, which means that if we needed to get information from right here, then we'd be able to get that information pretty easily because it's accessible if I knew where to look. Okay, if I try to find that information and it's down in the middle of the spool, there's no way. Like, there's no way I can get to it. Okay, and it's the exact same thing with DNA. The spool would be the chromosome form of DNA, and the idea of the chromosome form is it's not useful as far as finding the information on the DNA, but it's very useful in terms of organizing the DNA. And if you remember, we had to organize the genome and split the genome into different cells during mitosis. So what this takes care of is, if we have these chromosomes instead of chromatin, then then when we go to do mitosis, it's real easy to grab a spool. If you could imagine having a couple dozen spools of string, and if you took all the string off all the spools and just threw it on the floor, it'd be this crazy tangled mess. And then I said, hey, I need half those spools to go into another room. You would never be able to untangle them. You'd be like, forget it. Like, we're just going to trash these things and go get some new stuff. But if I had a couple dozen spools of fishing line and they were still on the spool, and I said, hey, I need half of those into the room next door, you just grab the different spools because they're organized. All that string is organized on these. So that's kind of the idea between a chromosome and chromatin, whether it's organized or not. Okay, and I'm going to get rid of these pictures here. Let me see other terms on this. Okay, I'm going to go to the next slide so that we can kind of kind of stay organized here. Okay, we're going to continue on this genome idea, but we're going to look a little bit more closely at one of our cells. If we looked at one of our cells, here we go, we have a whole lot of chromosomes in there, and so we'd have, I'm not going to draw them all, but we'd have 46, but let's just draw more. Okay, so we got a bunch of chromosomes in here. Bunch of chromosomes, which we haven't really introduced what they look like, but those are all chromosomes. It ends up that each of these chromosomes represents a set of information or a book. We have 46 books in our cells. 1, 2, 3, 4, 5, 6. So we have 7 here. I'm going to make it 8 just to round it off. So we'd have, this cell has 8 different books in it. Each book carries a different set of information. And so if we highlight this, so this one right here might carry the information for eye color and the shape of your nose and whether you have an aggressive personality or not. And this one is the information for whether you have hair on your knuckles and if you're able to curl your tongue or something like that. But they all have different sets of information. Okay, and all of these together make up your genome. Right? So we need to talk about some different shapes, some different parts of these chromosomes and what they represent as we're moving forward here. So I'm going to insert a picture, if I can. It's not letting me. Try double tapping, still won't let me. There we go. Hey, this is it. There's our picture. Hello, picture. This is a electron micrograph right here of an actual chromosome, and then over here are some diagrams of chromosomes. What I want to first highlight here is this is showing a chromosome. This is also showing a chromosome, even though they look different. The comparison here is this has that chromosome, this has the chromosome, and an exact copy of the chromosome. Okay? If you remember before mitosis, we have to take our information, make exact copies of it, and then we can separate those copies and put them into the new cells. All right? So use some terminology here. We already talked about genome, chromatin, chromosome, and chromatid. Okay, sorry, this one was chromatin. Chromosome and chromatid is what we're seeing here. Chromatid is, chromatid would be one side of a chromosome. That's a chromatid, that's a chromatid. They're called sister chromatids if they're on the same chromosome. And technically this is also a chromatid. Okay, it just hasn't been copied yet. Sister chromatids are genetically identical, 100% the same, so that when they get separated during mitosis, we end up having the exact information on both sides. Okay, for us, we would have 46 of these, okay, right before mitosis starts, so that each cell can get 46 of these in them, in all the daughter cells. Again, each of these is a book of information, and we just made a copy of the textbook, if you will, or a copy of the book of information over to here. Okay. The centromere, next term, centromere is this location right here where the chromosome, the two chromatids, are kind of pulled together pretty tightly and they indent. They call it kind of like a waist. I think it like you have a waist where you put a belt around. It's kind of like that. There's a series of proteins that connect our two chromatids together right through here. All whole length, they're connecting them. But in this area, they connect them a little bit more tightly, and so it produces this centromere in the central region. Okay. Other terms really quickly. Somatic cells versus gametes. We will find that in humans, we have some cells, almost all of ours, that have chromosomes, and we have 46 chromosomes. Okay. There's other cells that have chromosomes that look like this. We have 23 of those chromosomes. These are the somatic cells right there. The one that has 23, those are called gametes, either sperm or egg or gametes. Every cell in your body looks like these somatic cells, with the exception of a very few. And then the gametes would just be sperm and egg, depending on what it is that you produce. What was I going to say here? Within that 46 here, this 46 chromosomes, 23 of those you got from your mom, 23 you got from your dad. And they each have different sets of information, although they have the same types of information. You got some hair color information from mom, some hair color information from dad. We'll talk about that a little bit more later. But so that's somatic cell and gametes, and then finally the mitosis and cytokinesis thing. Mitosis technically is just taking the chromosomes and putting them on opposite sides of the cell. That's mitosis. Cytokinesis then is actually splitting the cell in half, and I actually have to cut this video off right now, or else it won't let me publish it because it'll be over 15 minutes. So I'm going to stop, and we'll see you in class.